Hey you fellas, Nick here. Don't mind me, I'm just reading this phone book. I'm looking for someone with ideas. It can get pretty hard to make a lot of those after a while, so, I'm looking for someone to call. Hopefully one of them has an idea that I can gush about for like, I don't know, 6-7 minutes. 10? Mitosis? I love mitosis, sure I'll talk about it. Mitosis is difficult to explain to people because, everyone's been through it at some point in their life. You see, living things don't grow by becoming one big cell. They're a lot like a bag of Cheez-Its. Instead of having one big Cheez-It, you get a bag full of tiny Cheez-Its. I promise this makes sense, just give me a second. The same principle applies to cells. It's faster to move cells around if you have little things because it means more surface area in the same amount of space. This is the idea of cell growth, where cells grow by populating, not actually becoming bigger. Now, how does it know how to do any of this? Well, through the use of the chromosomes that are inside of it. Chromosomes have a bunch of genetic info, and as the cells grow by dividing, they start passing over copies of it. You see, these identical copies being made of the nucleus and the chromosomes inside of it, that's mitosis. When the cell actually splits into two separate beings, that's cytokinesis. Allow me to demonstrate. Welcome to what the losers in my family call a kitchen, and what I call the Mitosis Mansion 3000. The patent's pending, so it's not totally legal yet, but it'll do the job. You see, the FDA won't let me cook food anymore after 2018's little incident, so I'm gonna have to figure some other way to explain mitosis while also using food. Like lying. As long as I don't tell the FDA that I'm actually making food, then they'll be fine with it as long as they don't find out. The only issue is that I was planning to use a chef's hat. I can't do that now, because if I did, the FDA would find out I'm using food, so I have to improvise. This'll probably do. Mitosis begins as interphase. This contains pretty much all the preparation needed to get cells ready for mitosis. The first phase of mitosis is the growth phase. This is when the cells grow and make sure that they have enough room to commit mitosis. To represent that, I'm going to be taking this salami. I'm going to be trying to grow it, so just a little bit of a little bit of that, and we'll see if it grows. So I'm back. I didn't read the fine print on the back of this. It clearly says not for growing. So that was a waste. Let's just go on to the synthesis phase, man. Next is the synthesis phase. Once you have all your salami and it's all grown, then it starts to duplicate through the process of DNA replication. So to represent this, I got all my salami duplicated. The second growth phase is pretty much like the first growth phase, but better because first is the worst and second is the best. But hold up, they have to go through some checkpoints to make sure that they aren't, bad cells. If every cell could just go through without it being checked to make sure it's actually good, I look like this. To make sure it's ready for mitosis, there's a wide variety of checkpoints inside of interphase. The first one is in the first growth phase, to make sure the cell's big enough to split into two. If not, it stops growing entirely and enters G0. You see, most cells, because they aren't dividing, actually are in G0, but if it is big enough, then it goes into S phase. S phase doesn't actually have its own checkpoint. That's integrated into the second growth phase. That one checks for DNA replication going well, and also for the second phase of growth being good enough. Now we can finally get into the first 4 phases of mitosis. First is prophase, where all the messy, idiot, stupid, dumb DNA gets organized into cool, awesome, smart, funny chromosomes. Taking these pieces of DNA does take a long time, hence why this area takes so much time out of mitosis. The next phase is metaphase, where the chromosomes decide now's a good time to organize in a single file line. These things called centrioles let loose their spindles, and they attach themselves to the midpoint of the chromosomes. This is named the centromere. The third and last checkpoint is stationed here, which makes sure the spindles actually did their job right and attached to the chromosomes all well and good. Once that centromere splits, it creates 2 halves that get pulled in opposite directions by the spindles to each side of the cell. This is anaphase, and it's pretty much where the cells actually start dividing. Last comes telophase, where the spindles break off and a nucleus forms around chromosomes on each side. You'll also start to see pinching. Now, after this is the finale of the cell cycle, which is cytokinesis. However, cytokinesis is either after telophase or part of it. Either way, this is when you see true separation. Well, I successfully mitosis, but this isn't what I'm supposed to do. I should have made something that's not food, and I made food. If the FDA finds out that I'm doing this stuff, They're gonna kill me, man. I can't do that. Maybe I should call it a day on the Mitosis mansion. Well, I need to throw my hat in the ring on the Mitosis mansion. I can't do this anymore. Instead, I'll be repurposing the hat into an advice column. You know, if you need advice with anything in your life that starts with M and ends with mitosis. Dear Nick, why do I have to mitosis? I've always hated this stuff since I was in the 2nd grade. The larger a cell is, the less efficient it becomes, so cell division is required to keep a bunch of smaller cells around. As your body develops and grows, new cells need to constantly take the place of old cells. Dear Nick, is there anything connecting cancer to mitosis? Cancer is pretty much when cell division gets worked into overdrive, causing huge clumps of cells that reproduce way too fast for the body. You see, proto-oncogenes are the things that help cells grow, and when mutated, they go way too fast. Then the tumor suppressor gene also gets mutated. What does it do? Take a guess. This gene pretty much makes sure that any damaged DNA doesn't spread, that cells don't leave S phase, all that kind of stuff. Basically, it's one big security camera. When it gets mutated, it's like taking a camera, taking a garbage bin, and doing that. When one or both mutate the cell division process, it causes the cells to divide so rapidly that tumors can begin to form. Dear Nick, is there anything connecting DNA replication to mitosis? Yes, DNA replication is involved in mitosis. These two are very much connected. I checked the facts. Like I said before, DNA replication is part of S phase, and without it, DNA would definitely be split wrong. Now, you can probably guess from the name, but DNA replication is pretty simple and, you know, replicates the DNA. Dear Nick, are there things that can harm mitosis? One of the major things that can affect it negatively is chemotherapy, a type of radiation. Chemo basically takes all the cells that divide really fast, which is literally what cancer cells are, and destroys them. That pretty much stops mitosis dead in its tracks no matter what you do. It's harmful radiation that kills cells like that, but it can also kill good cells, like for example hair. Since chemo is only targeting the fast dividing cells and not just actual cancer cells, it can't tell the difference and just kills everything that divides fast. Another thing harmed by it is various disorders in chromosomes. Take trisomy 21 for example, aka Down syndrome. The extra chromosome on pair 21 causes an imbalance in DNA in cells, as extra data is now being duplicated around your body. Dear Nick, how much useless information do you know about mitosis? Enough to make a top 5. Number 5. Endoreduplication is a rare type of mitosis where cytokinesis just doesn't happen. That means you get 2 nuclei in the same cell. Number 4. Mitosis is known as equational division sometimes because the amount of chromosomes is equal. You start with 1 cell, 23 chromosomes, you end up with 2 cells, and 23 chromosomes each. Number 3, mitosis was first observed by Walter Fleming in 1879, but was observed incorrectly 40 years before that by Carl Nagelli as some abnormalities in some dead cells. Number 2, Fleming used the Greek root word mitos to represent mitosis because the chromosomes bundling together during mitosis look like threads, which is what the word mitos means. Number 1, there is a secret phase of mitosis not many people know about. It's called prometaphase. This is when the nucleus around the chromosome starts to break down, and the chromosomes have fully condensed, meaning that the spindles can attach to them and start slowly arranging them around. Pretty cool, right? I've been reading that list to bed every 3 years because, you know, just in case. Dear Nick, where do you see mitosis evolving as the Earth changes with things like global warming or climate change? Well, this is completely a full-on opinion moment, but I don't see it changing that much. While yeah, it's a super complex system and it has a lot of parts to it, I don't see, you know, that one thing causing it to change a lot. I mean, historically, mitosis hasn't really changed that much anyways. Well, that should be all of them. Where did this come from? Dear Nick, not a question, just a cease and desist. This is the FDA. Stop making food. The jig is up. Well, now the FDA is off my back, I've decided to quit illegal cooking. I've now converted to professional headline reader. Sandwich stock rises 3,000% this week. Oh, come on!